bio book out. We have a lot of work to do. So let me actually stretch my screen like this. We want to quickly finish this bio syllabus. It's actually, yeah, the chances are increasing the more we push every day. I'm sure you can see that. Guys, can you all see that? And you do a lot of questions. So it's gonna be nice this weekend because I've been preparing for that. For a weekend of questions. So that's good. From 17 to 21, just doing questions together from year to year to year to year. And I give you some, I do some, I give you some, I do some. The way we do everything, that's the way we're just going to be doing. Are you ready for that? Huh? Are you ready for that, Lusika? Are you ready for the weekend? For a weekend of questions. Can you all hear me? Okay, good. Well done. Let me see. Ooh. Wow. Let me share my screen before we get started with the lesson. I want to ask you a question, guys. Look at this. I mean, this is saying only 4 people turned in. What happened for bio? You be having work from school or what? Tell me the truth. What is going on? KG, Laura, why didn't you turn in? Did you have work from school or something like that? Or Letty, will you be having work from school? It would be a lot. Like you couldn't do the questions. Yes, Misha, I see your hand is up. Tell me, what's up? Mm. 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 Mm-hmm. Okay. Okay. Okay, that makes sense. All right, fair point. That's what I wanted to know because I wanted to know. I know that when only 4 people turn in, it's definitely not a people problem. Okay, it's a system problem. When the results, it's just like, let's say people. This is like if this was a pass rate, this is nearly 1 out of 10. So that's a 10% pass rate, which means there's something wrong with the system. So okay, I'll adjust. Don't worry, I understand that. So at least your tutor has common sense, right? Okay, no problem. I will extend the time and allow you guys to submit during the weekend. If you can submit, please do. Consistency is so important. Um, I'll think about it. I'll think about everything so that I find the best way to do all this. Right. But the most important thing is that we move on. So let's move on. What I wanted to do, the reason why I was giving you bits and pieces of work was that I wanted to make an end of week report for all the students. Okay. Because now I have like an admin team. So I was working with them to put an end of week report for you guys. But so I wanted them to like get you a report every end of week, how you would have done that week. But it's fine. I'll extend the days so that you guys can submit maybe by Saturday morning or something like that, so you can do it Friday night because it's not a lot of questions. Biotech genetic engineering. So what were we talking about? Well, we talked about the usefulness of bacteria, why scientists love bacteria, and we looked at the products of biotechnology. I don't think we finished, but yes, now we wanted to go on to look at fermenters, and genetic modification. So this is biotechnology. We have to quickly go through this. We shouldn't make it hard. Once it looks hard or like there's a lot of things, then we know that there is something that we are not doing right. Okay? Are we together? So let's just quickly bam let's quickly move through everything and be as thorough as we can. Last time we said last time we were looking at the last thing that I wrote. Let me see. Right. It was okay. This is the usefulness. Where's the table? I gave you a table. Okay, here is the table. And then I was explaining them now. I was giving you the information. So these are the 6 products of biotechnology. Now I was just giving you one by one what each does. So let's continue with that. I talked about biofuels. We know that biotechnology is used to make biofuels, bread, fruit juice. I explained that fruit juice part. Washing powder, it's biological washing powder, not just washing powder, but biological washing powder. So don't just say washing powder, okay? Be more precise. Then, so let me say it. Let me fix that. Biological washing powder. Lactose-free milk and penicillin. Lactose-free milk for people who are lactose intolerant, which means they don't have anything to produce lactase in them. So they need milk which does not have lactose. Because if lactose stays as lactose in the body, then somebody becomes ill. Okay, they become ill. They start to feel headaches and tired and everything like that. So because we need to help them not to go through that, biotechnology gives them lactose-free milk. Let me explain to people so that I'm not just yapping like a parrot. Bio has 21 topics. We are doing topic number 21. Okay, we have 21 topics to make sure we know. We have done 17 up to 20. We are doing topic number 21. And then we're going to do a weekend of questions. So those who were not there, make sure during the weekend you're there when we do a quick rundown. And then when we do questions, it will be very helpful for you. Make sure. 17 to 21. Those are the topics we did. We're going to be doing questions for that over the weekend. And I'll be making everybody focus on the main things from each topic so that you get the marks you need. Thank you. So right now we're just trying to quickly finish topic number 21, and then we can see. So what we are doing is we said that topic 21, biotechnology and genetic engineering, I put the parts and I showed you the 6 parts that you have to know. And so far we did part number 1 and part number 2. That's the one we are doing. So we'll try to do 2, 3, and 4 today. And then we'll have 5 and 6 to cover in the next lesson. So let's quickly go through it. I'm about to start. Products of biotechnology was the part number 2. Biotechnology is when you are using microorganisms like bacteria, like fungi, very small things that usually people don't talk about in life, okay? It's not the things that you come across every day like a plant or an animal or something. It's bacteria and fungi. These are very small things. So if you use them to produce things, then that's biotechnology. If you use them to make biofuels, if you use them to make bread, if you use them to make fruit juice, if you use them to make washing powder, if you use them to make lactose-free milk, you have to know this. You have to keep going through this over and over again. I can't remember it whilst you can't. Well, you can't have me remembering and then you failing to, okay? Surely you can just go through this until you guys remember this. It will be very, very good. Now, let's quickly go through the other things. I was saying, after bread, the third thing, let me make a would be fruit juice. Everybody, right? Fruit juice. Let's see how biotechnology helps to make that fruit juice, which is called 100% fruit juice. Right? So, fruit juice, which is made 100% from the fruits. Really? Yes. They don't really add water, not much. 100% from fruits. How do they do it? Are you ready? All right. So, write this. Not all juice comes out when squeezed. This is very true, guys. Trust me. Not all the juice comes out from the fruits when you squeeze them. We put 10 apples and got very few juice, very little juice, very little juice. So whilst we had put it in the, I don't know what you call it, is it the crusher or whatever, that machine, and it was shocking. What? How? But this is the reason. Not all fruit juice comes out from the fruit when you squeeze the fruit. So the solution to this would be to add pectinase enzyme. And this pectinase, we know that it is made from microorganisms. That's why we are studying it in biotechnology. So the secret would be to add pectinase, right? Pectinase breaks down pectin in plant cell walls. Okay, in the cell walls of plants, there's something called pectin, and pectinase breaks it down, okay, in plant cell walls, okay, to produce more juice. To produce more juice. Okay, to produce more juice. Okay, and clearer juice. Are we together? Hello? Yes. So write this. This is important. Pectinase is produced by microorganisms. So this is where the biotechnology comes in. We use microorganisms, bacteria, fungi, whatever, to produce this pectinase. And this pectinase, we add it to fruits. Once you add it, that same fruit is going to release much more. Really? Yes. So you get much more juice? Yes. Without doing it, you're going to get nothing, my friend. You're going to waste. There is no way you make a lot of fruit juice. You'll be just wasting. So that's biotechnology, okay? Which is used in making fruit juice. Biotechnology is also used in making biological washing powders. Biological washing powders. Right. Biological washing powders contain enzymes. Okay? Biological washing powders contain enzymes that break down fat, protein, and food stains. Biological washing powders have enzymes, and these enzymes break down fat, these enzymes break down protein, these enzymes break down food stains. Are we together? Okay? They help to, okay, or they are suitable for delicate clothes. They're suitable for delicate clothes. Okay? As they help save energy and get clothes clean faster. Okay? They help to save energy and get clothes clean faster. Are we together? Yes. Now, the enzymes are produced biologically. So it is part of biotechnology. The enzymes you find in biological washing powder. Okay? So you can say, what do you mean when you say they are produced biologically? I'm just saying the enzymes in biological washing powders are produced by bacteria or fungi grown in fermenters. Okay? So they are then, okay, they, when I say they, I mean enzymes. The enzymes are then extracted and purified and companies put them into washing powder. So you no longer have the bacteria, the fungi that actually did that, but because they were produced this way, we still count them as biotechnology. Okay? Good. So just like for pectinase, okay? We know that it was the same thing for pectinase. Don't forget, we did not say, hello, we did not say that uh, pectinase is the microorganism. No, but pectinase was produced by microorganisms. So microorganisms produce pectinase. Scientists grow the microorganisms. Okay, scientists grow the microorganism. That's biotech. That's biotech there. We are growing the microorganisms to use them for this. Scientists grow the microorganisms that produce the pectinase. Okay, they grow the microorganisms. We know what they do. They're going to do some genetic engineering there. Put a gene into these microorganisms. Then they grow them inside fermenters, and the microorganisms begin to produce pectinase. Then pectinase is collected and added to fruit. Okay? Are we together? Hello? And added to the fruit. And added to the fruit. So these are products that are made by microorganisms. It's different from bread making. It's different from biofuels. Bread making and biofuels, we were using the organism itself. We were using the yeast itself to make the biofuel. We were using the yeast itself to make the bread. But here we are not using the bacteria to make the fruit juice. Bacteria don't make fruit juice. Bacteria is nowhere near the fruit juice. Make no mistake there. You are not going to put bacteria into fruit juice. What are you saying? We know that you put yeast into bread. We know that you put yeast into biofuel. Yeast is a fungi. It's a microorganism. You put it directly into these things. So for the first ones, these products of biotech, you were putting the microorganism itself into the biofuel or into the bread, but you are not putting the microorganism into the fruit juice. This is now different. You are going to use the microorganism to produce something that you will then put into the fruit juice. For bread, we put the yeast in the bread, and it started to do its anaerobic respiration there inside the bread, using the bread, the flour. And that is where you had the carbon dioxide making and stuff like that, which will cause the dough to rise. And when the dough rises, now we can have the bread being soft. It is soft. You can cut it nicely and stuff like that. Okay, so yeast was added to bread. Yes. Here, yeast is added into the fuel to produce this biofuel. Yes. You take your sugar cane or whatever, you add yeast there. But don't talk about, there's nothing which is going to be added here. They'll ask you, what microorganism do you add to fruit juice? Nothing. So what do you add to fruit juice? Pectinase. Then they'll start laughing at you to say, you don't know what you're talking about. So pectinase, how is that biotechnology? You say, it's still biotechnology. Because the pectinase was produced by bacteria. Bacteria is not being added to fruit juice. It's a little bit different now. But still, bacteria produced pectinase. Pectinase was added to the fruit to break down pectin and let fruit juice come out. Biological washing powder, we are not going to add the bacteria into biological washing powder. No. We are going to take the bacteria and produce enzymes. Then we are going to take those enzymes and add them to biological washing powders or to washing powders. And once you add them, now that washing powder can break down fat and protein and food stains from clothes. They can remove food stains. How are the food stains going? It's the enzymes that were produced by bacteria. So this is still biotechnology. We are using bacteria to make these enzymes. So this is a bit of a two-step process. Are we together, everybody? This is easy, right? For penicillin, for bread, for biofuel, we are using the microorganism itself. But for these other ones, lactose-free milk, there's no microorganism you add there. You take microorganisms somewhere separate, you make lactase, then you bring the lactase into the milk. All right. So let's talk about it. Lactose-free milk. Okay, we are just left with two more. Okay, lactose free milk. This is the next. This is the next. Some people cannot digest lactose. So if it goes into them, it will make them ill because their bodies cannot digest it. Am I being heard by my students? Students, there are people who cannot digest lactose. Scientists add, okay, lactase into the milk. Okay? Are we together? Okay, some people cannot digest lactose, e.g., lactose in milk. So scientists will add lactase into the milk. Lactase breaks down lactose into simpler sugars. Lactase will break down lactose into simpler sugars. The result from that is lactose free milk. Now these people can no longer be made sick by that milk. Okay? Are we together? Yes. Well done. Right. Now, skip a line and say, how is that biotechnology? Lactase is an enzyme. Scientists obtain or produce lactase from microorganisms. Are we together? So you are not putting the microorganism into the milk carton. You make people sick. You can't do that. You are going to take the microorganism, produce something called lactase. You collect that lactase and purify it, then add it to milk. Okay? Are we together? The lactase enzyme is then collected and purified and added to milk. Okay? The enzyme then breaks the lactose in milk into simpler sugars, glucose and stuff. Okay? You find out if it's glucose and maltose or whatever. And result would be lactose free milk. Result would be lactose free milk. So there's biotechnology in that the lactase was not manufactured in a plant or in the sky or collected from a walking animal. No, it was produced from a microorganism. So that's biotechnology right there, man. That lactase is now taken to lactose and they treat the milk and it can no longer make people sick. So whether you are lactose tolerant or lactose intolerant, you are fine because there is no lactose there. So there is nothing to be scared of. That's a good thing. So now you don't have lactose intolerant people getting sick of milk because it no longer has the thing that makes them sick. They are now happy. We have treated the milk. Now it is lactose free. Are we together? Mm-hmm. Penicillin is the last thing. Now, this one is just like biofuels and bread. It's directly from the microorganism itself. The microorganism does not make an enzyme, then the enzyme help. No, 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 no. For penicillin, there is no enzyme, what. It's straight from the fungus. This one is actually made by the fungus itself. So just like the bread, we put yeast into the bread, and yeast worked on the bread. In the biofuel, we put it into the mixture where we wanted the fuel. Here we are putting it, we have what we call penicillium fungus. It is the one that will actually produce penicillin. Are we together? Mm-hmm. Penicillin is produced not by a plant. It's not produced by a plant. It's produced by penicillium fungus. So right there, that's why this is biotech. We are using a microorganism. Anytime we use a microorganism to do things, that's biotech there. You're not using a donkey. You're not using a tree. You're using fungus, a microorganism. Are we together? Penicillin kills bacteria. And stops the growth of bacteria. And stops bacterial growth. Are we together? Yes. It kills bacteria and stops bacterial growth. Penicillin. Right. We move. It is actually an antibiotic, that one of the first ones that was discovered. So I don't need to write. That's not anything that I need to write to say it was the first antibiotic discovered. That's nothing to do with biotech. It's not something that. But are we fine? Okay. So don't let me down. This one, there's not much to write because it's just direct from it. Straight from the microorganism. Right? We are using the microorganism directly. So, good. You don't have to do insulin at your level, do you? If we see where they says I should teach you about insulin production from a fungus, then I'll teach you because it's also made by bacteria. Insulin is made by bacteria, so I'll only talk about it if it's part of the syllabus. Can we now go to the next part? Where so that you see even how this penicillin is produced. Right. Part 3. Industrial fermenters. I'll show you the diagram. But let's talk about it first. An industrial fermenter is where we are going to be growing all these things, these microorganisms. So an industrial fermenter is a large container used to grow microorganisms under controlled conditions. Under controlled conditions. So a large container used to grow microorganisms. Are we together? Scientists use fermenters to grow one, bacteria, two, fungi, three, penicillium, which is still a fungus, to be honest. And yeah, let's say all those things that you grow after you change their genes, those microorganisms. So you take a microorganism, you take one, right? A few, you change the genes in there, in a few, then you now grow them. Now we know that bacteria grow fast, so that's one of their advantages. So once we put them into fermenters, we expect very good growth there. So fermenters, they are controlled conditions that we use to grow the microorganisms. Don't make this complicated. What is complicated about this? Just a container where we are growing bacteria. A container where we are growing bacteria. And this container is good because we can control the temperature, pH, everything we are controlling. Okay, to make sure that these things are growing well and fast. That's your fermenter. That is a fermenter. Genetically modified. Why is it called a fermenter? Here's why I'm about to show you. I mean, everybody knows what fermentation is. There's no oxygen there. Right. Let's put conditions that must be controlled in a fermenter. Okay. So a fermenter can grow bacteria, can grow fungi, can grow penicillium. It's a large container which can grow these, and it grows them under controlled conditions. Bacteria, fungi, and genetically modified things, and other genetically container under controlled conditions. Let's see what we mean. You can't just tell us under controlled conditions. What do you mean when you say which conditions? Which conditions do we control in a fermenter? That is what I'm giving you right now. Conditions that must be controlled. Okay? What conditions? Which conditions are you talking about? How can you just say conditions? Which ones? The conditions that you control. Let's start with the easy ones. Temperature. Okay? Temperature. Because you are producing enzymes there. Those microorganisms that you're growing have enzymes there in the mixture. So come on now. You have to make sure that the temperature is great. So control the temperature, please. So temperature. So you need optimum. You need to control temperature for optimum enzyme activity. You need to control the pH for the same thing, for optimum enzyme activity. Can you see? You have to control the oxygen. You have to control the nutrients inside the fermenter. Okay? And you have to make sure it's clean. So you have to contain sterility. You have to make sure that there is no contamination. You have to sterilize. And the other thing which you should is the stirring. You need to stir. Why? To distribute oxygen and nutrients evenly. For sterility, I said you have to prevent contamination. You have to prevent contamination. Then you have to monitor the nutrients. Okay, you can. Why? Because you need growth. Okay, and you need respiration. Which respiration? Aerobic. So for oxygen, you need it for aerobic respiration. Right? These are conditions these are conditions that you will control. Okay? This makes fermenters useful because they allow one, large scale production of microorganisms or enzymes or whatever. Large scale production of microorganisms and their products. They allow controlled conditions. Faster growth in fermenters because conditions are being controlled. Okay? And then there is maximum product yield, which means the amount of product. You get the maximum amount of product. Thank you, fermenters. You are large, you are big, so we can do a large scale production of these things. And we can have controlled conditions. We can be controlling and fixing to make sure everything is perfect. Everything will be perfect inside these fermenters. You can get the maximum product. We know that if we want to make penicillium, we'll grow our penicillium fungus in here and we can be collecting the penicillin. But it's different when you want to do what? It's different when you want to make biological washing powders. You're not going to do that in a fermenter, obviously. Come on now. But you're going to collect the enzymes, then you're going to add to biological washing powders. Are we together? We'll do diagrams and see some questions that they like to give you from this that have diagrams. Okay, guys? All right. Let's go for this. Let me go for 4 and see if I'll be able to finish this. Genetic engineering now. Genetic modification. What are we doing? We are putting new genes into this bacteria. The gene controls the behavior of the cell. Gene is a set of instructions. We know that the complete set of instructions will be the DNA. But the gene will be the set of instructions that are responsible for one job. So they control the production of one particular protein or whatever. So when we are changing the gene, we are trying to change what it produces. We want it to produce one particular thing, which is whatever we put in there. If we want them to produce penicillin, or, right? If we want them to produce whatever else you want the bacteria to produce for you, the enzymes you put in washing powder, then, my friend, you have to put a new set of instructions, which is a new gene. And that is how we want to see how you do that. Everybody here knows you gotta do that. Everybody knows what a gene is. It's the brain box. And gene is responsible for one thing. So that's the one thing we're trying to change. Come on now. Right? Guys, are we on the same page? You're on the same page, of course. Right? I believe that. Now let's go. So let's see how we do it. Genetic modification. We have to do it. We want it to produce insulin so that we fight type 1 diabetes. So we can give people insulin in syringes that they can put every time they eat a meal containing starch. So genetic modification means changing the genetic material of an organism. Of course, it's going to be a microorganism that we're going to be changing. We change its genetic material. Okay, by inserting, removing, or altering genes. If we can put new genes or alter the ones that were in there or remove, we have done modification, we have modified, we have altered. To modify is to alter. So genetic modification, in this case that we're doing, it means altering genes. And then we will get what we call a GMO. You guys know GMOs. You've heard about them. Genetically modified organisms. Right? So let's talk about them. So genetic modification in this case it means altering genes. There are now new genes that were not in there. Right? Results in GMO, which is genetically modified modified organisms. Okay? And if you have modified it by putting genes that came from another species, then we will call that thing, let me write it. This genetically modified organism, if it got DNA from some other species, then we are going to call it now a transgenic organism. Trans-swapping genes, transgenic organism, which means organism that contains DNA from another species. Organism that contains DNA from another species. What we will see is, can we do that in plants? That's what we will see. Can we do that in plants? Where can we do that? We'll see that process, the process, because you must memorize it. Okay? Now, you see, this organism which has gotten a gene from another species is called transgenic. Now, a transgenic organism has got genes, is an organism that got genes from another species, which is another organism. Now, the genes it now has together, okay, we know that all your genes are called DNA, right? So a DNA that is a mixture of different genes from different organisms is called recombinant DNA. So I want you to write to say, this leads to recombinant DNA. Recombinant DNA. It is that the recombinant DNA is found in transgenic organisms. Transgenic organisms are organisms that have received genes from another species. So now their DNA contains genes from another organism, and their DNA is called a recombinant DNA. So transgenic organisms have recombinant DNA. Recombinant DNA, okay? They have recombinant DNA, which is DNA that has genes from different organisms. Are we together? Right. Okay. These are keywords. Don't let me down. Genetically modified organism, an organism that has, it's just a genetically modified, where you have altered the genes, okay? You have altered its genes. Whether you have removed one of its genes, or you have swapped one of the genes, or you have added a new gene, that's a genetically modified organism. You have modified those genes. Genetic modification process. Are we all on the same page, students? Scientists move a useful gene, like the gene that produces insulin, okay, from one organism to another. Thank you for knowing this. But that's not where we're going. Okay? This is called genetic modification. And you must memorize the process. You must know the process. How this happens. How do we take a useful gene from one organism and put it into another to alter? Of course, that's altering. But we don't take, genes that are not useful. We want the gene that produces insulin. How do we do that? It's 7 steps. So I want you to say genetic modification process. I want you to say the 7-step process. Okay? I should have written it somewhere nice. I don't know what I did there. 7-step process. Okay? And we'll see keywords there. Are you ready? What is a GMO? What does the word GMO stand for? What does GMO stand for? Genetically modified organism. What have we changed? What have we altered? We have altered the we have altered the genes. Okay. When we alter genes and we bring a foreign gene into something, what type of organism do we get? Transgenic. Transgenic. Says itself. Then a transgenic organism will have what type of DNA? A DNA that is made from genes from different organisms. It calls what? What is it called? Yes. I'm happy. You are pushing. Come on. Are you not proud of yourself? That's it. Good. Let's do the 7-step process now. Step 1. Step 1. The scientist must find the desired gene. Okay? What gene do you want? You have to find it. Remember, every gene has a location. I kind of said it in chapter 17. I don't know if you remember. Every gene has a location. So they should just know where it is located on your DNA strand. Then go and take it, that gene. Right. Now, step 2. They must cut the gene, okay, using what they call a restriction enzyme. Using a restriction enzyme. There are going to be keywords that I'm going to say. Restriction enzyme. This is a keyword here that I'm going to need to tell you. Okay? It is an enzyme that cuts DNA like scissors. It cuts DNA. And when it cuts the DNA, it creates what we call sticky ends. So it goes on DNA, it cuts it, creating what we call sticky ends. The ends are sticky. Okay, restriction enzyme cuts DNA. Okay, are we together? Maybe I should put it below this. Should I write this? Restriction enzyme. You can say enzyme that cuts DNA. Are you seeing? Creating sticky ends. Are we together? So cut the gene using restriction enzyme. Don't let me down. Do not let me down. Then step 3. Step 3 of this, what are we doing? We are altering the genes. So we have found the gene with that we want. Where are we going to put it? Are we going to put it into a fly? No. Are we going to put it into a butterfly? No. Into a duck? No. Into a horse? No. Into a donkey? No. Where are we going to put this that we are doing this? Into bacteria. We are taking it to bacteria. Okay? The microorganism. So we have identified where we are taking it from. We cut that gene using, not using a scissors. Restriction enzyme is a molecular scissors. It's a molecule that acts like a scissors that cuts DNA and it makes sticky ends. Now, I want you to say, cut the plasmid. We will now talk about this plasmid here. We'll talk about it. Cut the plasmid using the same restriction enzyme. You have to cut the plasmid using the very same restriction enzyme. Why? So that you are going to make matching sticky ends. So I want you to say here, as step 4, matching sticky ends are formed. So now our plasmid has a cut which matches the cut on the DNA, the gene we've cut, so they can fit into each other. The DNA and the gene is going to be joined into the plasmid. The gene will be put into the plasmid. Okay, like a puzzle, pieces fitting into. So I want you to say sticky ends. These things that we call sticky ends. I want you to say these are small letters of DNA that are exposed. So you can say small exposed bases that they should match. Okay, small exposed bases that allow DNA pieces to fit together like a puzzle. So if we can only learn this skill, scientists, if you can only know how to use this restriction enzyme, if you can only know how to use the same restriction enzyme on a plasmid, if you can only know how to find the desired gene, then welcome to biotechnology. You are going to be useful because you help do this process. So that's what you got to learn. Then you can now help. So matching sticky ends are formed. Step 5. Step 5. The DNA ligase joins the gene and the plasmid. Okay? Or joins the gene into the plasmid. The DNA ligase joins the gene into the plasmid. You say, sir, you've been telling me all these new words, these keywords, sticky ends, you told me what it was, restriction enzyme, you told me. What does DNA ligase say, which now joins the gene and the plasmid together? It's a glue. So I want you to say, it acts like a glue that joins DNA pieces together. So the good thing that we have is that bacteria cut the plasmid in bacteria, or you place it into bacteria. Let's just say cut the plasmid. Leave it like that. I don't want to be too specific. Cut the plasmid. The good thing that we have is that bacteria has plasmid. That's the best thing we have, because it will now be able to work inside the plasmid and produce whatever protein that you want. So if we go to step 6, you will now just say, insert this plasmid into the bacteria. So here we'll be dealing with 1. So you insert the plasmid into bacterium, and now that gene is now in bacteria. Really? Yes. So the bacteria will start doing what the gene was doing? Yeah. So if the gene was producing insulin inside your body, the insulin where? At the pancreas. Hey, Mr. Pancreas, you stopped. So what we'll do, we'll say, okay, give us that gene. We take that gene, we put it into bacteria. Guess what? It will not be stubborn like the pancreas. It will that bacteria freely produce insulin. So you can, whatever the protein it is, the enzyme. So that is what we'll be just doing. It will be making all those enzymes that we talked about. So bacterium produces the desired protein. We're together. Hello. Of genetically, examples of genetically modified, should I say crops? Organisms. Examples of some organisms that have been genetically modified, that have a new gene in them. Hmm? Genetic modification, changing the gene. You've seen the steps, you're not going to let me down. One is bacteria with the insulin gene. I've said it many, many times since the beginning of the lesson about this. I told you about bacteria that has the insulin gene. This is a real thing which helps produce insulin, which is saving lives. So this is not a joke right here. This is something very important. What does it do? Okay? The bacteria, what does it do? The purpose, the bacteria now, bacteria the bacteria produce insulin. Okay, they have been given this gene of insulin. What other example do we have where we have changed the gene of something? In crops, we have changed the gene to make crops that are insect resistant. We can't have crops that, a lot of them are killed by insects and the yield is very low. No. We can't have crops that are just killed by pests. We can make some crops that will kill those pests, the insect pests. So these crops, I want you to say the crops kill insect pests. They kill nuisanceable insects. The crops will kill these nuisanceable insects. Insect pests, which means insects we don't want. Okay? So you have less crop damage. Are we together? Right. You have less crop damage. Maybe let me put here to say advantage of this, less crop damage. The advantage of this, you have less crop damage. Okay? And you have you use less herbicides. Are we together? Or pesticides. Less pesticides used. Lower farming costs. Let me just say that. Now, disadvantages of this. The disadvantage of doing genetic modification to create insect resistant crops is what? Genes may spread to wild plants. Okay? And you can say farmers become dependent on companies. If a company has made a mistake somewhere, it will cost the whole thing. So you can't just do that. There are also some disadvantages. Okay? What if there is less yield? What if that crop does not produce as much? You went to buy this crop because it can kill pests. But what if it has side effects? which will affect you as a farmer. You have done 6 months of growing something or 4 months, and then when it comes out, it's not good. The yield is not good, but the pests were killed. What good does that do? Have you seen the disadvantage of that? Huh, students? Have you seen the disadvantage? Another one, which is the third example of a crop that has, I want you to say, herbicide resistant crop. Herbicide resistant crops. Right. Here we've seen crops that kill insects. Now here we are seeing herbicide resistant crops. Are we together? Right. I want you to say here, herbicide kills, or you can say here in brackets, weed killers. Weed killers. They kill, right? Herbicides are also called weed killers. They remove all the weeds, the undesired plants. Okay? Are we together? Herbicides, they eliminate unwanted vegetation. So what happens? We will do genetic modification to make sure the crop itself is herbicide resistant. Are we together? Here, actually, the advantage of this, did I write less herbicides used? Okay. Less herbicides used. Herbicides kill unwanted plants. Pesticides kill pests. So this plant, because it's killing its own pests, this insect resistant crop, the insect resistant crop is killing its own pests. Yeah? So we don't need pesticides, and we also don't need herbicides sometimes. Obviously, here, herbicide resistant crops, we are going to spray herbicides, and these crops won't be affected. Why would they not be affected? Because the gene we gave them is going to protect them. So we're going to have advantages. So let's first talk about this. Herbicide, these ones, they now survive crops. Okay? The crops, what happens when you do this? The crops now survive herbicide spraying. What is the advantage of them surviving herbicide spraying? You can now kill the weeds, and they survive. And weeds, the problem with weeds is they are competing for resources with the real plant. So you have the plant being thin because it's fighting. But once you kill the weed, now you have higher yields because this plant is getting all the food to itself. Are we together? Are we together, students? Okay. Disadvantage of using herbicide is this. These genes may spread to wild plants. Any gene you use to enhance your crops, what if it goes to the wrong plant? What if that happens? The other problem is you have possible growth concerns. And you can have reduced biodiversity because everything else beside your crop is going to die. So that's what happens. You can have reduced biodiversity. Okay, this is good. I'm happy with where we've gotten to. We don't have much more to cover, everybody. We'll do it in the next lesson, okay? The last part. But it's basically mostly summaries, okay? The most important things we've covered there. The 6 pillars, okay? We've talked about bacteria. They reproduce quickly. They have plasmids. And they can produce complex things as their benefits. We said it last time. And we talked about biotechnology, what it is, and the products that it makes. We talked about fermenters, to say they grow organisms in controlled environments. We said genetic modification, we will be adding or removing or altering genes. We talked about how to do it, how genetic engineering happens. The steps, the 7 steps, where you use a restriction enzyme to cut both your plasmid and your gene. You cut it out of the DNA. And this makes sticky ends that match. The DNA ligase then joins those sticky ends that match. Then the plasmid now has the gene in it. Then you place it into the bacteria and the bacteria begins to produce proteins. We didn't talk about how you can take it out and whatever. That's not important. So here you have put that. This is very important what you have done. Then lastly, we saw some examples. You know, there's a fourth example which you must know, which is called golden rice, by the way, of organisms that are genetically modified. We have bacteria within the insulin gene. We have insect-resistant crops. We have herbicide-resistant crops. We have golden rice. Everybody must know about golden rice, okay? You should go and research about golden rice. Why did they do the genetic modification? We'll finish it in the last lesson, but we are done basically. I'll just put a little bit of additions to things that I think you may need for the questions, and that I'll compile whilst preparing the questions for you guys that we are covering for the weekend. So make sure you're ready. We have to cover these topics that we have been doing. We have to go through a lot of questions. Inheritance. We'll talk about meiosis, mitosis, protein synthesis, Punnett squares, variation in selection. We'll talk about it. Natural selection versus artificial selection. What is variation? What does it come from? We'll talk about it. Then we'll talk about organisms and their environment and stuff. Then how humans impact the ecosystem. Then we'll talk about this chapter. And we'll be done. We'll do a lot of questions. A lot of questions. So, of course, tomorrow is maths and physics. But physics we'll try to finish again tomorrow. And we'll do the questions together. So less activity over the weekend, please, to make yourselves available for interesting questions. And of course, maths, we have a lot of work to do. We need to do a lot of maths together until you are able to do a lot of maths in the correct way. That's it for bio. We are switching to chem. Let me just mark the register first, and then we'll switch to chem. Give me a sec. Yes, hi. Okay. Let me share them. Once I stop sharing the screen, I'll share them into the group. No problem.